Well, hello everybody. Welcome back to Monday. Monday, this is day 7 of 11 days of biochemistry, and we're right where I want to be. So I see that some of you in Detroit there, in Columbus, it's raining up there. Rainy days and Mondays always get me down, but today you're not going to get down because we got a lot of high-yield stuff coming your way. You're going to be glad that it's raining up there because you have nowhere else to go but to be indoors and to listen to biochemistry. So anyway, we've got a lot of good stuff coming your way, a lot of high-yield stuff. You're going to be thankful that you came tonight because a lot of integration stuff with biochemistry with endocrinology, and you're going to see a lot of pathology as well tonight, especially later on in the evening. Okay, so where we're at is, so last week we spent a lot of time talking about genetics and molecular biology, talking about chromosomes and genes and RNA and this and that and all that kind of stuff. But what's that got to do with anything? And what's that got to do with life? And the idea, as we talked about on the last day on Saturday there, we talked about the idea is that in your body, you are a bag of chemicals. Even though you don't like to think about it, but you are a bag of chemicals. And in these chemicals, you have pathways to convert chemicals from making glucose and burning glucose and making fatty acids and burning fatty acids all in the same cell. And all these chemicals in one cell can go into different kinds of other chemicals. So the idea is in these metabolic pathways is but the rates of these chemical reactions are too slow. That's why you have enzymes encoded by genes in 46 chromosomes that make these reactions go faster here. And then, so we talked about the concept on Saturday of why you have enzymes in the first place. So again, to make the reactions go faster. And we talked about the concept that when when one enzyme is missing, then you got a major problem called pathology, because the rate is too slow of a particular reaction. And then if the rate is too slow, the substrate accumulates. And when the substrate accumulates, it spills into other metabolic pathways, causing havoc to the cell, and then the organ, and then the body suffers as a consequence here. So again, the concept of why we have enzymes in the first place. Now, chapter 9, that's where we're at right now, is the issue of why do we have hormones? And the idea, this is major integration with endocrinology. And the idea is, of the hormones that we have, there are two major classes, and one is the fat-soluble hormones or lipid-soluble hormones versus the water-soluble hormones. Now, the lipid-soluble hormones were already discussed. That was way back in molecular biology. All lipid hormones, they work at the gene level, whether it's cortisol, whether it's testosterone, estrogen, or whatever, they all work at the gene level. And as a result, because they work at the gene level, they work very slowly. Now, in this chapter, we're going to talk about how do the water-soluble hormones work. And because they're water-soluble by definition, water-soluble hormones, what that means is they cannot enter the cell because they're water soluble. They cannot enter the cell. They cannot go through the plasma membrane to come into a cell to turn on a particular pathway. So as a result, the hormone must bind to a receptor on the cell surface of a particular target cell, and then it signals. And then there's signal transduction that ultimately results in the turning on or turning off of a particular pathway. So that's what's going to be discussed in chapter 9, are 3 kinds of signal transduction pathways. Now, the first of the 3 is the signal transduction pathway that's mediated by glucagon and epinephrine. How does glucagon or epinephrine, how do they turn on and turn off pathways? The second of the 3 signal transduction pathways we're going to review in this chapter is how does nitric oxide as well as ANP, how do they signal vasodilation. You know nitric oxide causes vasodilation of smooth muscle, but how does it happen? How does it occur? It's all coming up in the second of the 3 signal transduction pathways. And then thirdly, we're going to talk about the signal transduction pathway that's mediated by insulin as well as growth factors. How does insulin or growth factor, when it binds to its point of view? Dr. Wilson, several weeks ago, and I guess it was in March when he started the course off, he talked about the physiology point of view of these hormones, but now the biochemistry point of view. So the first one is, again, you got to know this information of how these hormones work. So the first of the 3 signal transduction pathways is, how does glucagon, how does epinephrine, how do they mediate signal transduction? Okay, so now suppose we have a plasma membrane of a target cell for glucagon or epinephrine. Now, because glucagon is a polypeptide hormone, because epinephrine is a small water-soluble hormone, they cannot enter the cell to turn on a particular pathway. So as a result, these hormones must bind to a class receptor on the plasma membrane of the target cell. Now let me ask you this. What is the target class receptor? What's the target class receptor? Remember, lipid-soluble hormones. Let me ask you this. What do lipid-soluble hormones use? What class receptor do lipid-soluble hormones use? Let me see your answer. What do lipid-soluble hormones use? Not GRE. That's the GRE is the target DNA sequence. But it is, that's right, it's the zinc finger class of receptor. The zinc finger class of receptor, which is also classified as a specific transcription factor class. So again, they are specific transcription factors, but the class of receptors are called the zinc finger receptors. And then when they are turned on by the hormone, they go into the nucleus looking for a piece of DNA called the HRE, also called the GRE, those things we talked about earlier here. So those are response elements, but they are DNA. They're not the receptor. The receptor is the zinc finger class of receptor here. Okay. Now the urease, not CRE, not PPREs. Again, that is that's the target DNA. That's not the class of receptor here. Okay, now with these water-soluble hormones, what's the class of receptor here? With glucagon and epinephrine, what's the class of receptor? The class of receptor called? Let me see your answers here. What's the class of receptor? No. Let me say, there we go. And they're called the adrenergic class of receptor. Adrenergic class of receptors. Now, when you the epinephrine binds to the beta adrenergic receptor or the alpha adrenergic receptor class. Now, glucagon, you just don't use the word adrenergic, but it's the same principle. Now, so the idea is these are 7 transmembrane domain proteins. They're sometimes called the serpentine receptors because they span the membranes multiple times. But the idea is these are the adrenergic class of receptors here. And there are a variety of these important receptors. But let me stress as much as I can. So many medical students, when I ask, what is the class of receptor? So many students will say it's a G protein class of receptor. Never say that. The G protein is never ever a receptor. The G protein is never a receptor. Never. They're on the wrong side of the membranes. The G protein's on the inside of the cell, and the hormones on the outside of the cell. They never see each other. So the class receptor is never a G protein. The G protein may be coupled to the receptor, but it's not the receptor. So again, it's the receptor class is not a G protein. Never ever say G protein for the receptor class. It's the adrenergic class of receptor. Okay, here it comes. Now, when glucagon or epinephrine binds to the beta adrenergic receptor class, again, there's different kinds of beta receptors, beta 1, beta 2, beta 3. But when epinephrine binds and glucagon binds to the beta receptor, the receptor undergoes a conformational change. And then on the cytoplasmic side of the receptor, it then activates a trimeric G protein called GS. And trimeric meaning 3 subunits, alpha, beta, and gamma. And because it's a G protein, it means it binds GTP in order to work. By definition, any G protein means it binds GTP in order to work. All right, here comes. Which of the 3 subunits, you must know this, which of the 3 subunits, alpha, beta, or gamma, which is the one that binds the GTP after activation? That's right, it is the alpha subunit. So the alpha subunit binds GTP, and then it undergoes a conformational change, and then beta and gamma go away. And then alpha S of GS, for the stimulatory G protein, alpha S of GS is now active because beta and gamma go away and it's bound to GTP. And then in turn, it activates another enzyme in the inner leaflet of the plasma membrane, and this one's called adenyl cyclase. Now this enzyme, adenyl cyclase, on board exams, you might see it called adenyl cyclase, or you might see it called adenylate cyclase with an ATE ending, or you might see it called adenyl cyclase with 2 YLs. It's all the same enzyme, different names for the same thing. Now, what does adenyl cyclase do once it's activated by alpha S? What it does is it takes a little bit of ATP in the cytoplasm, takes off the 2 terminal phosphates, and then it makes as a product cyclic AMP as the product. Now, I'm going to show you, it's not in your book, so don't go looking for it. I'm going to show you the reaction catalyzed by, my wife will get it, sorry about that. I'm going to show you the reaction catalyzed by adenyl cyclase. Now, here's the reaction catalyzed by adenyl, because sometimes if you see a structure, you remember it you remember it better. If you see a structure, you think, oh yeah, now I see what's going on here. Here is the reaction catalyzed by adenyl cyclase. Here it is. So what happens is, so again, alpha subunit of GS will activate adenyl cyclase, and what it's going to do is going to take this molecule at the top, and then now if you were asked, what is this molecule at the top, or what is this molecule? Well, when you look at it and say, well, it's not cholesterol, I wonder what it is. So you look at it very carefully, and you notice it has a base, a sugar, and carbon 5' 3 phosphates. Base, sugar, phosphate. Oh, it's got to be a nucleotide triphosphate. So then which nucleotide triphosphate is it? You look at the top of the molecule, and you see 2 rings. You say, well, it's 2 rings, it's got to be a purine. So if it's either adenine or is it guanine? So then you look at it carefully, and you say to yourself, well, at the 12 o'clock position, is there an amino group there? And the answer is yes, there's an amino group there. And if there's an amino group on a purine at the 12 o'clock position, therefore it's got to be adenine. Adenine linked to ribose and 3 phosphates, that's ATP. Look how simple that was to figure that out. It wasn't hard, it wasn't anything magical. It was a matter of thinking about it. That's ATP at the top. It was a piece of cake figuring that out. See, that wasn't hard at all. Now, what does the enzyme do? Adenyl cyclase. What it does, it cleaves this bond right here. It cleaves this bond right here, right there, cleaves that bond right there, and splits off inorganic pyrophosphate. How often have we talked about that? Inorganic pyrophosphate being generated, so which is rapidly destroyed because it's toxic and it's high energy. But the idea is the remaining phosphate is then cyclized between carbon 5', which it's already on, and then cyclizes it with carbon 3'. So adenylcyclase now puts the phosphate also on carbon 3', also on carbon 5'. Now, because the phosphate is linked to carbon 5', it's a monoester linkage there. Because the phosphate's on carbon 3' of the ribose as well, it's a monoester linkage there. Mono here, mono there, together. It's a phosphodiester is the product. And the product is called 3', 5', cyclic, because the phosphate's in the cyclic nature. 3', 5', cyclic AMP as the product. Abbreviated cyclic AMP, but it's more appropriately called 3', 5' cyclic AMP. But remember, the phosphate's linked to two different carbons. It's in a cyclic nature. That's why it's called a phosphodiester linkage. The phosphate's linked to it's linked to two different carbons of the ribose. Please keep that in mind in just a few minutes. So again, there's reasons why I'm showing the structure here, but the product is cyclic AMP as a product. Should you remember these details? No, nobody's going to ask you to draw the structure of cyclic AMP, but you should know what it means. Why? Because you're going to see the significance in a few minutes. You'll see why it's important to understand what's going on without the gory details. So the details, I'm not going to ask you for the structure, but you should know what 3', 5' cyclic AMP means. All right. Okay, so now once cyclic AMP is made, there it is. It can then, before we talk more about it here, let's go back to the other situation, is that when epinephrine, in physiology, when epinephrine binds to the alpha-2 adrenergic receptor, again located on other tissues, but when epinephrine binds to the alpha-2 adrenergic receptor, that receptor becomes activated. It then stimulates on the inner leaflet another G protein called GI, inhibitory G protein. And then alpha I of GI binds GTP, beta and gamma go away. And then alpha I of GI then inhibits adenylocyclase, because that's what the I stands for, inhibitory G protein. And then alpha I of GI inhibits adenylocyclase from making cyclic AMP. Please keep that in mind, because at the end of the chapter, we're going to be talking about how does pertussis toxin work. And you must know the issue that alpha I inhibits adenylocyclase from making cyclic AMP. Now, coming back to the beta receptor. When glucagon or epinephrine binds to the beta receptor, it then triggers a GS to bind GTP. Alpha S then is now activated. It activates adenylocyclase to synthesize cyclic AMP. Now, once cyclic AMP is made, it turns on so again, the alpha S stimulates adenylocyclase, alpha I inhibits adenylocyclase. Again, please remember the physiology in where these receptors are located, as in terms of which G protein is being stimulated, whether it's GS or GI here. Now, when beta receptor is triggered and GS is stimulated, and then cyclic AMP is made, then the cyclic AMP then turns on a major enzyme you've got to know, and this enzyme is called protein kinase A. Now, let me stress here, from now on, now until Friday when I'm done, anytime I highlight an enzyme in the red box, anytime I highlight an enzyme in the red box, especially once we get to metabolism, automatic, high yield. Sometimes I forget to say it, but if I forget to tell you that, if it's highlighted in the red box, you got to know that enzyme. Now, what does protein kinase A do when it's stimulated by cyclic AMP? Because it's a kinase, it means it's going to use the terminal phosphate of ATP in the cytoplasm and use that phosphate to phosphorylate what? The first word says proteins. So what's going to happen is some enzymes are then phosphorylated, and by phosphorylation of certain enzymes, some enzymes are turned on and some enzymes are turned off by phosphorylation. It depends what enzyme we're talking about, whether the enzyme is on or off by phosphorylation. One more time. So again, protein kinase A was phosphorylate certain enzymes, and by doing so, some enzymes are turned on in certain pathways, other pathways by phosphorylation of enzymes. Some enzymes are turned off by phosphorylation by protein kinase A. All right? Okay, now what's coming up here is a question for you. The cyclic AMP, once it's made by the adenylocyclase, has two fates. Two fates. One fate is to activate protein kinase A. Let me see. Now, what's the other fate not shown on this figure? What's the other fate of cyclic AMP? Let me see your answer here. Not shown on this figure. What's the other fate of cyclic AMP? What else can happen to cyclic AMP in a cell besides activation of protein kinase A? What other fate is it? Anybody? Inhibits GI? No. Good try. Good try. No. Good try. DNA? No. Yeah, there we go. See it? There we go. There we go. And that is to be degraded, to be turned over. And the name of the enzyme that turns over, that degrades the cyclic AMP is a major enzyme. You talked about it in physiology, you're talking about it now, and you're going to talk about it in pharmacology as well. And that's called phosphodiesterase. Phosphodiesterase. Remember that I showed you the structure on purpose of cyclic AMP. Remember that 3' 5' cyclic AMP? I showed you because I knew I was going to talk about this. Because what the phosphodiesterase does, it cleaves the phosphate from carbon 3', and the phosphate, it remains on carbon 5'. That's why the product is called 5' AMP as the product, because the phosphodiesterase cleaves the phosphate from carbon 3'. The phosphate remains on carbon 5', but it's no longer a cyclic molecule, and it's destroyed. In other words, it cannot activate protein kinase A. It can't activate protein kinase A. All right, now another item here. Another item. And that is, you always, so again, cyclic AMP has two fates. One fate is to activate protein kinase A to cause certain enzymes to be phosphorylated, turning some enzymes on or off. And the other fate of cyclic AMP is to be destroyed. Now, another question for you here is that you always hear about cyclic AMP as the second messenger, right? You always hear about, it's the second messenger. And it is the second messenger. All right, now here comes a question for you. What is the, if that's the second messenger, what's the first messenger? Did you ever ask that question? Did you ever ask, what's the first messenger if that's the second messenger? What's the first messenger? Not the G protein, not the G protein, not the G protein. Not the receptor either. A lot of you are getting the right answer. Not the receptor complex. No. The primary message, it's the hormone. The hormone is the primary message being sent by the body. After all, what is a hormone in the big scheme of life? Whether it's a water-soluble or a fat-soluble hormone. What is a hormone in the big picture of life? It is a message. It's a message being sent by the body. Go tell some organ or tissue in the organ to go turn on or turn off a particular pathway. Now, the lipid-soluble hormones, they're they are a primary message, but the lipid-soluble hormones can get into the cell. They can go through the plasma membrane. They can come into the cell, and then once they come into the cell, they can bind to the receptor and then bind to the DNA and turn on gene expression. They don't need a second messenger. Lipid-soluble hormones do not need a second messenger because they can go right through the membrane and turn on, turn off pathways directly. Whereas water-soluble hormones, they are the primary message as well. But because they cannot enter the cell to turn on pathways directly, they must transduce, signal transduction, the words. They must transduce the signal that the body wants accomplished of turning on and turning off certain pathways here. So again, the hormone is the primary message, and then transduce the message to a second messenger inside the cell to go on and turn off particular pathways. Always try to understand the bigger picture. What's going on here? All right, now another item here is this enzyme, the phosphodiesterase, is always working, and it's not regulated. It's always working. It's sometimes called constitutive. In other words, it's always destroying cyclic AMP. Always destroying cyclic AMP. Well, if it's always destroying cyclic AMP, you got to ask, well, then how do you ever turn on this protein kinase A? And the answer is, and it comes up in biochemistry underpinning of the physiology. How do you activate the protein kinase A if cyclic AMP is being destroyed all the time? You got to secrete the hormone. When you secrete the hormone, it binds to the receptor. It activates the GS, which in turn activates adenocyclase to make so much cyclic AMP. You make so much cyclic AMP, you overwhelm the ability of the phosphodiesterase to remove it. By overwhelming it, you then activate protein kinase A to phosphorylate certain enzymes, turning some enzymes on and off. You simply make so much more cyclic AMP, you overwhelm the ability of the phosphodiesterase to remove it. So again, it's a competition. So the idea is you're secreting the hormone, you're making so much cyclic AMP, and cyclic AMP has two fates. One is to be destroyed, which is always happening, and the other is to activate the protein kinase A. And because in the presence of the hormone, you make so much cyclic AMP, you overwhelm the phosphodiesterase's ability to destroy it. That's how you turn on this part of the pathway down at the bottom. How do you turn this off? You stop secreting the hormone. When you stop secreting the hormone, you stop activating the receptor, you stop activating the GS, you stop activating adenocyclase, and it's because you're not activating adenocyclase anymore, any cyclic AMP that happens to be around continues to be destroyed, so the levels drop and drop and drop and drop because it's no longer being made anymore. Eventually there'll be no more cyclic AMP, and you stop activating protein kinase A. It's a competition. So again, one more time. How do you ever turn it on? You secrete the hormone, you activate cyclic AMP, overwhelm the ability of the phosphodiesterase to remove it. And therefore you activate protein kinase A. How do you ever turn this off? You stop secreting the hormone. And by stop secreting the hormone, you're no more activating adenocyclase, no more cyclic AMP, and what cyclic AMP is around is no longer being remade, and its levels drop to nothing. There it is. There it is. That's the biochemistry underpinning of the physiology. It's a competition that goes on there. Amazing how this happens. Amazing how this happens here. All right, now on the right side of the figure, more endocrinology. Under conditions in which vasopressin is secreted or epinephrine binds to the alpha-1 adrenergic receptor. Let me ask you this. In physiology, what is the downstream purpose triggering alpha-1 adrenergic receptor. What is the downstream purpose of triggering alpha-1? Let me see your answer here. That's right. To increase smooth muscle contraction, right? See, that's what they tell you in physiology. See, in physiology, they talk about all these hormones, and they talk about what pathways are being turned on, off, what's happening in terms of organ, tissue, increased smooth muscle contraction, blah blah blah. But that's all the end product, the final product, the final situation that occurred by that hormone. But in between is the biochemistry. What caused the vasoconstriction that went on, and by secreting epinephrine binding to the alpha-1. So the idea is if you think about this a minute, is that vasoconstriction, because you're doing smooth muscle contraction, that means ATP is being consumed in a muscle cell. And because ATP is being consumed, the muscle cell needs glucose to burn, to make ATP, to do muscle contraction. So that's what's going on here by the alpha-1 adrenergic receptor, is the generation of fuel to burn to make ATP to do muscle contraction. So how was this accomplished? So when the alpha-1 adrenergic receptor is triggered, yet another trimeric G protein is triggered in turn. This one's called GQ. Alpha Q of GQ binds GTP. Alpha Q in turn then activates yet another enzyme in the inner leaflet of the plasma membrane called phospholipase C. And what does this enzyme do? It looks for a small molecule in the membrane called PIP2. It's a small lipid in the membrane, and what it does, it does a hatchet job. It cuts it right down the middle. PIP2 is cut right down the middle, generating two fragments. One fragment is called PIP2, other fragment is called IP3. One of these fragments, IP3, will then go to the ER and then open up a channel in the ER to allow calcium to come out of the ER, and in the muscle that's called the SCR, it's called the smooth endoplasmic reticulum. But the idea is the calcium then comes out, and then along with diacylglycerol together, then stimulate yet another kinase called protein kinase C. And then protein kinase C then results in the phosphorylation of certain enzymes in the cell, in the smooth muscle cell here. So the concept here is that these three, diacylglycerol and IP3 and calcium, all three of them together, diacylglycerol, IP3, and calcium, all three together, are referred to as second messengers. Second messengers. And because the hormone is the primary message, the first messenger, and these are the second messengers because they're being stimulated by signal transduction. And therefore, these are the second messengers. Now, another item here, and that is, if they were to ask you on board exams, what are the direct activators of protein kinase C? You answer by saying diacylglycerol and calcium, not IP3. IP3 is not a direct activator of protein kinase C. It's diacylglycerol and calcium together stimulate protein kinase C. IP3 is like indirect, but not a direct activator of protein kinase C. Okay, now the idea here is now on the bottom right hand part of the figure is we're going to try to figure out what pathway is being turned on here. Again, you know, you're sending out these hormones, and the pathway is going to be turned on. But the idea here is, let me ask you this: in physiology, is you talk about when you secrete these hormones. You say, well, because of increase in smooth muscle contraction, or you say here on the left side, you secrete epinephrine because you're exercising, things like that. But as simple as I can state, what is the main reason why you secrete these hormones? And here comes the reason. Why are you secreting these hormones? It's because the blood glucose is dropping. If you're exercising, as soon as you do skeletal muscle contraction, the ATP is being consumed, the muscle cell removes glucose out of the blood to burn that glucose to make ATP. And because blood glucose drops, you send out hormones called epinephrine. Or if you're sleeping in bed, you start secreting glucagon. Same thing with smooth muscle contraction. So the idea is that you're sending out these hormones because the blood glucose is dropping. So now, here comes the question. On the right side over here, what pathway makes sense to you is being turned on? Not gene expression. No, good try. What pathway is being turned on? Not the Krebs cycle. Good try. And the answer is glycogen breakdown. The idea is, as soon as the blood glucose is dropping out of the blood, the body responds by secreting hormones, and it's going to tell the liver, for example, go turn on glycogen breakdown to maintain blood glucose, because we don't want to die. The blood glucose is going to, remember this, we talked about this on Saturday, that if the blood glucose dropped below 3.5 millimolar, because of that zero order transporter we talked about, GLUT1 and 3, if it dropped below 3.5 millimolar, that means less glucose in the blood, and therefore less glucose entering the brain. The brain's going to suffer coma time, and that's bad news. So the idea is the body's going to say, hey, start breaking down glycogen in not only in the liver but also in the muscle and so on. So you start breaking down glycogen, which begins in seconds or minutes here. And again, it just makes sense. You can start rapidly breaking down glycogen within seconds or minutes. But it turns out that the amount of glycogen in the cell, in your body, the amount of glycogen in the human body is limited. Not now, but later on, I'm going to ask you, how long does your glycogen last? Don't answer right now. Don't answer. But somewhere along the line, I'm going to ask you, how long does your glycogen last? You do not have an infinite supply of glycogen. You do not have an infinite supply of glycogen, but it will run out sometime. And so the body says, okay, well, we know it's going to run out. So therefore, we got to start kicking in another pathway because the blood glucose will drop and will die. So the body says we got to kick in another pathway while the glycogen is running out. And sure enough, what happens is this same enzyme in the liver, protein kinase A, what it's going to do is going to look for a specific transcription factor called Krebs, which is normally inactive. And then by phosphorylating it, it's now active. And again, Krebs stands for cyclic AMP. Notice it's responding to the cyclic AMP. Cyclic AMP, response element binding protein. And when it's phosphorylated, it's active. It then goes into the nucleus of the hepatocytes, looks for a piece of DNA called the CRE, which is an enhancer region, which is and it's an enhancer for a major gene in the pathway called gluconeogenesis. And sure enough, that gene will be turned on by RNA polymerase, and then as a result, the pathway of gluconeogenesis is turned on, and you start synthesizing glucose. So the concept here is that this Krebs protein is, we already talked about this. We have already reviewed this earlier. Where did we first talk about this? Where did we first talk about turning on gene expression for gluconeogenesis? And you're all going to say right here, page 78. Page 78. This is where, just look at that page. You'll see this part of the figure right here has already been reviewed. How you're turning on gluconeogenesis at the gene level here. All right, now the concept here is, now what happens, so you start turning on gluconeogenesis, and because it's taking hours, because it's being done at the gene level here. All right, and I got a question for you here. Is not a question per se, but the idea is, what happens is a lot of students think what the body is doing is that when the blood glucose drops, the body first breaks down glycogen and then kicks in gluconeogenesis. In other words, sequentially. You first break down glycogen and then you kick in gluconeogenesis. There's no way that can happen sequentially. You'd be dead if it happened sequentially, where you first did glycogen breakdown and then you did gluconeogenesis. You'd be dead. Why would you be dead? Look at it. Look at it. Because glycogen, when it runs out, if the body says, well, okay, now I got to kick in gluconeogenesis, because it takes hours to kick in. When glycogen is gone, the blood glucose continues to drop and drop. And then you say, well, okay, I got to kick in gluconeogenesis because it takes hours. The blood glucose will drop to more and more and more. You'd be in a coma and you'd be dead. It cannot happen sequentially. You'd be dead. What I'm driving at is that this is happening simultaneously, concurrently. While you are breaking down glycogen, simultaneously you're kicking in gluconeogenesis, such that when glycogen is gone, and it will be gone, gluconeogenesis better be at its maximum, keeping me and you alive. And it will be at its maximum when glycogen is gone, keeping me and you alive. And how can this occur simultaneously? Notice the same enzyme, protein kinase A, is kicking in glycogen breakdown and simultaneously kicking in gluconeogenesis. Beautiful, beautiful how this works. While you are breaking down glycogen, simultaneously you're kicking in gluconeogenesis. You must understand they are occurring at the same time. And you must understand you'd be dead if it occurred sequentially, not simultaneously. You can't just memorize that. You have to understand why it has to occur simultaneously here. They go on together. Now, one more item here. One more item. And that is okay, everybody with me here. You must understand this. This is part of endocrinology and biochemistry here. Now I got a little oral vignette here. Oral vignette. Suppose they say to you that tomorrow morning you wake up, and when you wake up, you measure your blood glucose, and you find out it's a certain value. And then the day after that, when you wake up, before you measure your blood glucose, you go to your kitchen and you make and drink a cup of black coffee. No sugar, no cream in that coffee. And then after you drink that coffee, then you measure your blood glucose and you find out, wow, look at this. My blood glucose is higher than it was the day before when I didn't drink that coffee. How do you explain that? Obviously has something to do with this page, right? Has something to do with this page. Why did your blood, so here comes the question. Why did your blood glucose rise when you drank that coffee? Or I can change one word instead of saying coffee. Why did your blood glucose rise when you drank that tea? That black tea. No sugar, no cream in that tea, yet your blood glucose would rise. What's it got to do with this figure? Okay, that's right. There's something in that coffee. There's something in that coffee or tea. And what is it in pharmacology? What's that class of molecules called? The class of molecules. I know it's caffeine, but what's the class of molecule called? What's the class of molecule called? Stimulants, agonists, that's too, there we go, now we're getting close. Semenanthe, that's right. They're called the methylxanthines. The methylxanthines in pharmacology. And what do the methylxanthines do? Why did your blood glucose rise? And the answer is right here. Because the methylxanthines, what they do is they inhibit the phosphodiesterase. That's what they do. And so now why did your blood glucose rise? Because here it is. Because when you drink that coffee, when you wake up in the morning after sleeping, you're secreting glucagon. Glucagon is activating its receptor, activating GS, activating aminocyclase to make cyclic AMP. And a portion of the cyclic AMP, when you wake up in the morning, is supposed to be inhibited by the phosphodiesterase. But now if you inhibit its destruction, what happens to the cyclic AMP? It starts to rise. One more time. And so if it starts to rise, more protein kinase A, more glycogen breakdown, more gluconeogenesis, and your blood glucose would rise. You'd be hyperglycemic. Okay. One more time. By drinking coffee or tea, you're inhibiting the phosphodiesterase when you wake up in the morning. And therefore, some of that cyclic AMP is supposed to be destroyed, but now it's not being destroyed because the PDE is being inhibited. Therefore, cyclic AMP levels rise higher than normal. If it rises higher than normal, more protein kinase A, therefore more glycogen breakdown, and therefore more gluconeogenesis. That's why you become hyperglycemic. It makes sense. Now, if it doesn't make sense, find out why. It must make sense to you. If it doesn't make sense, don't be stubborn. Find out why. Use the Q&A pod. Dr. Prashad and Dr. Mahad, they'd be happy to answer the question, but it must make sense. You should be able to verbalize. Why is it when I drink a cup of coffee with no sugar in it, yet my blood glucose would rise? You should be able to verbalize the answer. Major integration of biochemistry with physiology and pharmacology here. All right. Okay, now what's coming up next? That's right. Kitsia. That's right. It makes sense. Eri. That's right. There we go. It makes sense. No excuses. If it doesn't make sense, find out why. I want you to know why. Here. You must know why. Now what's coming up next is the second of the three major signal transduction pathways, also discussed in physio and certainly going to be discussed in pharmacology. Now this is the issue of signal transduction to cause vasodilation. Now on the left, now suppose this represents a smooth muscle cell. Under conditions of distension, when the person secretes the hormone ANF. Now, by the way, you might hear it called ANF or you might hear it called ANP, but it's one and the same. This is the atrial natriuretic factor or peptide, same thing. So ANF or ANP is one and the same. It's a hormone that's being secreted under conditions of distension. Now, when the hormone is secreted, it's going to bind to its receptor. Now, notice here the receptor is not a serpentine receptor, not a 7 transmembrane domain. It spans the membrane one time. But when the hormone binds to its receptor, then the receptor in the smooth muscle cell becomes activated. And then on the cytoplasmic side, it'll take a little bit of GTP and then convert it by taking off 2 phosphates as 3', 5' cyclic GMP as the product. So again, in the smooth muscle cells, you're generating cyclic GMP inside the cell by the action of the hormone via its receptor. Okay, now on the right side of the figure, major integration with physiology and pharm. Under conditions in which you secrete vasodilators, what they're going to do is they're going to turn on a major enzyme in the vascular endothelial cells called nitric oxide synthase. Now, what does this enzyme do? It tells you what it does. It synthesizes nitric oxide using the substrate arginine. We talked about this on Saturday. One of the nitrogens of arginine is cleaved off. It's then made in nitric oxide as the product here. All right, now before we move on the upper right-hand side of the page, here comes the pharmacology. Now get ready with your responses. Under what conditions do you secrete nitroprusside? Under what conditions do you administer? What pathological or clinical problem do you administer nitroprusside to treat? Well, it's mostly used for hypertension purposes. Hypertension. Now, nitroglycerin would be used for angina. As when people said angina, so most often nitroglycerin would be used for the angina situation. What about isosorbide dinitrate? Dinitrate also would be used for angina. That's right, Marcelo. That's right, for angina purposes. Now notice here that these three pharmacological drugs are all nitrogen-containing drugs. So when they are administered pharmacologically, biochemically, they are then converted to nitric oxide here. So again, at the upper right-hand part of the page, look at the major integration of physiology on the left that Dr. Wilson talked about. On the right side, pharmacology that Dr. Harris will talk about. And I'm talking about regarding nitric oxide here. Major integration of three disciplines. Now, once nitric oxide is generated, fortunately, it's permeable to the membrane. There's no receptor for it. Fortunately, because its half-life is seconds. It sticks around only for a few seconds. It goes right through the membrane and looks for an enzyme called guanyl cyclase. And what does guanyl cyclase do? What does it do? Just like adenyl cyclase on the previous page, guanyl cyclase in the smooth muscle cell takes a little bit of GTP, takes off the two terminal phosphates, and makes 3' 5' cyclic GMP as the product. Notice here, on the left side, how ANF administration generates cyclic AMP, and so does nitric oxide generation, also generates cyclic GMP. Now, once cyclic GMP, let me finish off this figure, then we'll take the break. Once cyclic GMP is formed, it turns on a major kinase called protein kinase G. Here, now before we move on, now don't get these second messengers, because cyclic GMP is yet another second messenger. Don't get these second messengers and what kinase is being turned on confused. It's not that hard. Here it is. Here it is. This is what you must know. All you need to know is cyclic AMP turns on protein kinase A. Cyclic GMP turns on protein kinase G. And calcium turns on protein kinase C. Which second messenger is turning on which kinase? Okay, not hard. Not hard. All right. Okay, so again, you understand the concept here. You must understand the concept. Which second messenger is turning on which kinase? Who's zooming who? All right, now to finish this figure off. Now, there's a little dilemma which I would rather have you figure out first. Because notice here, a kinase is being turned on. Now, a lot of students won't get this right, so I want you to try to think about this. A kinase is being turned on, yet vasodilation is the outcome. So now we got to try to figure out, well, if a kinase is being turned on, that must mean, therefore, by smooth muscle that causes contraction. Let me see the answer. What's the name of the protein that causes contraction? What's the name of the protein that causes contraction in vascular smooth muscle? Chagrin, a little bit more detail. A little bit more detail. Name of the protein causes contraction. Not myosin. Which part of myosin? And it's called, here comes the answer. And it's called myosin light chain. myosin light chain causes contraction. Okay, but now let me ask you this. Does myosin light chain cause contraction in the phosphorylated state or in the dephosphorylated state? Let me see your answer. Does myosin light chain cause contraction in the phosphorylated state or in the dephosphorylated state? Protein kinase G to cause the vasodilation. There we go. Ahmad, anybody else out there? That's right. Without looking at Ahmad's answer, Fanny, that's right. Everybody, that's right. That's right. That's right. And the answer is, it's got to be a phosphatase, right? It just makes sense here. So now here it is. I'm going to say a sentence to you right now that sounds very convoluted. Very convoluted. But listen carefully, because this is exactly what happens. The straightforward biochemistry events. Here it comes. Here it comes. Ready? Everybody ready? I'm going to tell you the biochemistry of what's happening. Here it comes. The activated protein kinase G causes phosphorylation of a phosphatase. The phosphorylated phosphatase is now active. The phosphorylated phosphatase then causes dephosphorylation of the phosphorylated myosin light chain. The dephosphorulated myosin light chain cannot cause contraction. Therefore, relaxation occurs. Now, I know I said that very rapidly on purpose. I didn't stay up last night practicing that words. You can say the same thing as me. I'm going to say it one more time a lot slower. But you can say the same thing as me by just verbalizing the answer several times. Now, here it is. This is the sequence of events much slower the second time. Here it is. Now just look at the bottom of the figure and watch what happens. The activated protein kinase G causes phosphorylation of a phosphatase. The phosphorylated phosphatase is now active. The phosphorylated phosphatase then causes dephosphorylation of phosphorylated myosin light chain. The dephosphorylated myosin light chain cannot cause contraction. Therefore, relaxation occurs. Just like that. Just like that. Now again, while it's fresh in your mind, later on tonight or whatever, just practice the same verbiage. Look at this, then if you think you mastered it, look away and verbalize the same answer. Just verbalize the answer. Just try it on your own and you'll get to the same answer. You can't stare at this and expect to be able to spit it back later on a board exams. You have to practice this several times, and then you'll be just like me, just like that. Okay, just try it out. Just try it out. Okay, now one more item here. Then we'll take the break, and that is the major integration with pharmacology. Now, in pharmacology, at the break, or tonight on TV. If I go watch a basketball game, what happens is I go watch a basketball game on TV, the NBA, after we're done tonight, and then there'll be some commercials on TV, and in these commercials on TV, there might be a man and a woman sitting in the bathtub, and they're smiling at each other, and then a commercial will say, when the time is right. Okay, and now what's the name of that drug that's has something to do with this page? That's right. That's right. It is the sildenafil, Viagra, and so on. So now, what do they got to do with this page? And it has to do with the idea of the cyclic AMP. Now, remember on the previous page, cyclic AMP is always destroyed by a phosphodiesterase. Same thing here. The cyclic GMP is always being destroyed by yet another phosphodiesterase in the smooth muscle cells. And again, this phosphodiesterase is always working, always destroying cyclic GMP. And how do you ever turn this on? You secrete nitric oxide or you secrete ANF. You overwhelm the ability of the phosphodiesterase to remove it. And how do you ever turn it off? You stop secreting the hormones. And once you stop secreting the hormones, then cyclic GMP is no longer being made, and the phosphodiesterase will destroy it. But now the idea is, if somebody takes Viagra, then what the drug does, it inhibits the phosphodiesterase, so the cyclic GMP is not being destroyed. And as you know, the cyclic GMP, if it's not being destroyed, the levels start to rise. And when it starts to rise, you know the big picture in more ways than one. But you know where I'm driving at here. You know the rest of the story with that stuff there. But there it is. That is the idea. That's the target of the phosphodiesterase. All right, now let me ask you this again. So the idea is by inhibiting the phosphodiesterase, the cyclic GMP is no longer being destroyed. And if it's no longer being destroyed, look at all that relaxation, because it activates protein kinase G, activates the phosphatase. The myosin light chain is always dephosphorylated, causing all that relaxation. And you know where that's going on, all that relaxation that goes on. All right, one more item about this. On those commercials on board exam, on those commercials on TV, if you listen very carefully, the announcer will say that anybody taking a drug like sildenafil should not, they might say anybody taking an antihypertensive drug should not be taking sildenafil. No, because you could die. Now, why is that? Why is it bad or could be lethal by taking a hypertensive drug at the same time taking Viagra? Why could that kill you? That's right. Just look at the biochemistry explains it. There it is in front of you. Because if you take a hypertensive drug like nitroprusside and you generate so much nitric oxide, generating so much cyclic GMP, and a portion of it is supposed to be destroyed by the phosphodiesterase. But if not, if you're inhibiting the phosphodiesterase with sildenafil, then the cyclic GMP is so high, and therefore so much relaxation, the blood pressure, the smooth muscle blood pressure then would drop too severely, and you could die under those circumstances here. There it is, how the biochemistry explains the pharmacology. Everybody with me? Everybody with me? It makes sense. And if it doesn't make sense, find out why. All right, so ask. Okay, so right now it's 4 minutes after the hour. I'm sorry I went a little bit, but I wanted to finish off this figure. So let's take a 10-minute break, and then I will be back. See you shortly.